The overall goal of this procedure is to generate long-term cultures of adult mouse spermatogonial stem and progenitor cells, or SSCs. This is accomplished by first preparing mitotically inactivated feeder cells. Next, a testicular cell suspension is prepared by enzymatic dissociation of adult testis tissue. Then, the testicular cells are plated on top of feeder cells to allow SSC colonies to form. The final step of the procedure is to manually pick putative SSC colonies for subculturing in small culture wells. Ultimately, long-term self-renewal and expansion of SSCs over many passages in vitro can be confirmed by immunostaining for broadly expressed markers of germ cells, or more specifically of SSCs, and by functional analysis using SSC transplantation in vivo.
Generally, individuals new to this method will struggle because the identification of small SSC colonies by phase microscopy is extremely challenging.
Visual demonstration of this method is critical as the colony picking steps are difficult to learn, because it may be difficult to see SSC colonies against a complex background of other cell types that are present in the early stages of the cultures.
To begin, prepare feeder cells by culturing JK1 cells in a 100 mm cell culture dish with 10 milliliters of filter-sterilized feeder growth medium. When the culture reaches 95% confluence,